Hi, my name is Lala Bimanan, and today we're going to learn about DNA sequencing next generation. So here are a few of the topics we're going to cover. What is DNA sequencing? How did DNA sequencing next gen originate? How is it used? What are its limitations? And what is the future of next gen? To start off, we're going to go over what is DNA sequencing. Well, to figure that out, we have to look at Frederick Sanger and the Sanger method. Sanger sequencing, also known as chain termination method, was developed by the English biochemist Frederick Sanger and his colleagues in 1977. This method is designed for determining the sequence of nucleotide bases in a piece of DNA. Sanger sequencing with 99.99% base accuracy is considered the gold standard for validating DNA sequences, including those already sequenced through next-generation sequencing. Sanger sequencing was used in the Human Genome Project to determine the sequences of relatively small fragments of human DNA. These fragments were used to assemble larger DNA fragments and eventually entire chromosomes. Here's a quick glance at the Sanger method. As you can see, it has many steps, and because of that, everything is done by hand and extremely tedious. It wasn't until the Human Genome Project that the world of DNA sequencing got a little shook up. The Human Genome Project was one of the great feats of exploration in history. Rather than an outward exploration of the planet or the cosmos, the Human Genome Project was an inward voyage of discovery led by an international team of researchers looking to sequence and map all of the genes, together known as a genome, of members of our species, Homo sapiens. Beginning on October 1st, 1990, and completed in April 2003, the HGP gave us the ability for the first time to read nature's complete genetic blueprint for building a human being. The project didn't come without its controversies, and almost like a biology arms race, was the catalyst to promoting next-gen DNA sequencing. But here's a little background information. In the mid-1980s, some geneticists proposed a daring project to sequence the human genome. That meant figuring out the exact order of the 3 billion chemical pairs that make up human DNA. Sequencing the human genome could help scientists understand how our bodies work and help doctors diagnose, treat, and prevent certain diseases. The Human Genome Project launched in 1990 with scientists around the world collaborating on the research. They worked slowly and methodically, trying to produce the most accurate information possible. By 1991, one of these scientists, Craig Venter, became fed up with the HGP's slow pace. He challenged them to move faster and started his own company to compete with the Human Genome Project. Racing neck to neck, the two organizations reached their goal years ahead of schedule. But the challenge also led to a bitter public argument, especially over who could use the sequence and how. Venter's point of view was pretty straightforward. As he once said, I think scientists who claimed that they were going to do this public program also started to believe it was their birthright to get all this money and take the next several decades to do this project. I think the scientists were around this public effort because it was a massive effort to get billions of dollars in committed funding. This was the first time this has ever happened in biology. Well, the pace wasn't moving quick enough for Venter. He believed that shotgun sequencing was the fastest and most effective way to get useful human genome data. The method was rejected by the Human Genome Project, however, since some geneticists felt it would not be accurate enough for the genome, as complicated as a human's, and it would be logistically more difficult and that it would cost significantly more. Venter had opposing views, though. He viewed the slow pace of progress in the Human Genome Project as an opportunity to continue his interest in patenting genes. So he thought funding some private sector to start Celera Genomics. The company planned to profit from their work by creating genomic data to which users could subscribe for a fee. The goal consequently put pressure on the public genome program and spurred several groups to redouble their efforts to produce a full sequence. Venter's effort won him renown as his team and Celera Corporation shared credit for sequencing the first draft human genome with the publicly funded Human Genome Project. So what is shotgun sequencing that Celera helped to kind of usher in? Well, in 1981, Gene Myers, a current professor of computer science at Berkeley, graduated with a PhD in computer sciences from the University of Colorado and joined the faculty of the University of Arizona. While working at the University of Arizona, Myers worked with a geneticist named Jim Weber from Wisconsin in the field of bioinformatics. They came up with a revolutionary approach of large-scale sequencing projects such as the Human Genome Project. Their approach was called whole genome shotgun sequencing. Rather than the traditional method of separating the genome into BACs and sequencing each individual BAC via BAC and shotgun sequencing, Myers proposed the idea of breaking many copies of the entire genome into different sized pieces and sequencing those pieces in a special way. When this idea was proposed to the publicly funded Human Genome Project, however, it was met with great doubts. Many people doubted the ability of Myers' technique to sequence the genome, for they believed it would be prone to errors. But Craig Venter, the president of Celera at the time, saw potential in Myers' proposal and signed him on as the vice president of informatics research to test his approach. So this is how Celera really ushered in next-generation DNA sequencing. Upon the establishment of the Genome Project at Celera in 1998, the company purchased and connected 700 CPUs and 70 terabytes of hard drive space. This computing system was established to run the initial test of their algorithm code, which was to sequence the genome of the Drosophila fruit fly with a 13-fold coverage of the genome successfully in 1999. The most surprising thing about this approach was that it succeeded in coding the algorithm and sequencing 120 megabase paired genome of the fruit fly to that extent of completeness in just 11 months. Myers then modified the process so that the whole genome shotgun sequencing process would make a 5-fold coverage of the human genome, as he believed it would be adequate to provide a complete sequence of the human genome. In addition, Venter purchased 4 supercomputers referred to as a gene matcher from a company called Parcell Inc. Parcell Inc., a company that typically produces computers for government agencies such as the NSA, created this machine specifically for matching character strings, such as putting together sequences of DNA like a puzzle. It was composed of 7,000 processors arranged to perform over 1,000 times faster than any Pentium computer. With this new technology, on September 8, 1999, Celera began its sequencing of the human genome using this approach and completed the first assembly of the whole human genome in June 17, 2000, only 9 months after the project began. As you can see on the right bottom half of your screen is a picture of the Parcell Inc. computers. So with all of that background information, we can still ask, what is next-generation sequencing? Well, it's a massively parallel or deep sequencing related that describes a DNA sequence technology that has revolutionized genomic research. Using NGS, an entire human genome can be sequenced within a single day. In contrast, the previous Sanger sequencing technology used to decipher the human genome required over a decade to deliver the final draft. Although in genome research NGS has mostly superseded conventional Sanger sequencing, it has not yet translated into routine clinical practice. Here are a few pictures of some machines that are used today. Well, even though this technology is great, it does still come with some limitations. The main disadvantage in clinical setting is putting in place the required infrastructure, such as computer capacity and storage, and also the personnel expertise required to comprehensively analyze and interpret the subsequent data. In addition, the volume of data needs to be managed skillfully to extract the clinically important information in a clear and robust interface. So what does the future hold? Well, the global next-generation sequencing market is expected to reach $10.35 billion by 2025. With such a rapid and dramatic expansion and the ability to easily, inexpensively, safely, and accurately sequence variable lengths of nucleic acids in humans, plants, and microorganisms, it should be no surprise that next-generation sequencing protocols have been and are still being developed for everything from public health to food safety to plant biology. The future of next-gen sequencing of DNA and RNA is extremely bright, with applications extending as far as science's imaginations. It is not an exaggeration to say that the development of next-generation sequencing will always be viewed as a major breakthrough in the progress of biological sciences. Thanks for listening. Here are the references for my presentation.